last project that I did in the lab. And so this seems like kind of a silly idea of a brick wall in a seizure. But in order for you guys to understand what I'm talking about, I think we need to go through a couple of big ideas first. So someone tell me, what do you guys already know about seizures? What have you heard before?
I think you can assume not much.
Okay, cool. So in general, I feel like most people have heard of epilepsy and they know that seizures are a neurological disorder. And people usually think of seizures and they think of someone falling to the ground and shaking, right? And in a clinical setting, what a seizure is actually represented by is abnormal electrical activity. And so usually you can get a sense of what this looks like from a clinical perspective by recording using electrodes either on the scalp or inside on the cortex. So this is called electrocorticography or ECoG. And so up here at the top, what we see, oh, there we go, is a trace, and it's going to be in time. And what we see is that there's some point where all of the sudden, the activity seems to grow in amplitude, right? And it gets really big and really fast. And if we were to really zoom in on this, we'd see oscillations. And so this is typically what clinicians see that they identify and say, ah, okay, this is a seizure, right? They see this big, noisy, loud thing. But what's happening here, this is a pretty low resolution, right? We're covering a lot of the cortex at a time when we look at this sort of a recording. And so one of the things in our lab and our collaborator, Kathy Siobhan at Columbia, has looked at is what's really happening under the surface here. What are the neurons doing? Because the thing to remember is that this ECoG, these big traces, really only show us what the whole population is doing, and it's pretty low frequency activity that we're representing. So the way that we went about exploring this, and so this was work actually from Kathy's group, is that along with those ECoG electrodes, which are the numbered white ones, they inserted a microelectrode array. And so you can see that's this square right down here. And that is about the same size, but you get 96 electrodes, so 10 by 10 without the corners, microelectrodes. So all of a sudden, not only can you record the low frequency activity, but you can actually get neuronal activity during the seizure. So you can see what are the neurons firing, what does that firing look like? And what's really interesting is if you break that down, there was two parts to the seizure. There was a core where the neuronal firing was in sync with those oscillations. It seemed to be bursting, right? And seemed to be very phase locked. And they found that specifically, there wasn't any inhibition in this little bubble, right? And outside of this, there were neurons that didn't really look like they had anything to do with that activity whatsoever. And so this is what they call the penumbra. And over the course of the seizure, you can see that there's this wave front of very, very high activity that's slowly propagating outwards, recruiting neurons into that core. So all of this is well and good, but I just wanted to take a minute to explain this before we get into how this relates to filters and brick walls. So the important thing to take away here is that there's this spiking activity that may or may not actually relate to the low activity that we're seeing on this ECoG. Any questions so far? Cool. So the second thing that I want us to talk about before we really jump into how they're related is where this idea of a brick wall filter or a brick wall even comes from. So is today the first day that they are learning about filters?
Sorry. Yeah, no, it's it's kind of the end, towards the end of the standard lectures. We're going to talk about the Kalman next session.
Oh, cool. So have they heard about?
Know a little bit about the unit impulse response associated with an ideal filter.
Amazing. Okay. So then I will keep this brief and you guys can answer my questions instead. Challenge. Okay. So what we see here, and this should possibly look familiar if you've gone through the book, is this idea that you can have a unit impulse, right? A delta unit impulse here. And that if you put that through an ideal filter, you get out something that is called a sinc function. And you'll notice that it's oscillating in both directions. Oh, my bad. There we go. So can anyone tell me what is unique about this sinc function? What is it doing that we wouldn't expect a filter to do? Remember, this is where our unit was, right in the middle. And this is in terms of time. So this is equal to 0. So what kind of time is this? Time before or after our unit impulse?
Before.
Yeah, so this time that's before, we see that even though we put this through a filter, there seems to be a response. That sinc function is showing effects before time equals 0. So this is what we say is non-causal, right? So because we see this effect, and this is why usually we talk about how ideal filters don't actually exist in the wild, right? You can't have something that's non-causal. But if we look at, this is just a little clue, these are our experimental results. They look pretty similar to what we see in our ideal filter situation. So the question was, can we actually describe the effects of what we're seeing in terms of this BrickWall filter? So let's go back to the idea of that ECoG. We said that our ECoG recordings up here were made up of oscillations, and they really show us what the population is doing. And so if we took this a step further and we actually compute the power spectrum, we see that there seems to be one main dominant frequency that's coming out. And can you guys tell me, how do you find a power spectrum? Just see what everyone's learned. No, never mind. Okay. But the interesting thing was that we have this one dominant frequency. And part of my work before we even got to this project was looking at how the spikes that we could record from that microelectrode array related to those ECoG recordings, those low frequency oscillations. And so the way that we did this was to do spike triggered averaging. And so really what that was saying was, if you were to line up your spikes and your oscillations, for every spike, you select an epoch of your ECoG, and then you could average across all of these to see if there's any sort of a consistent relationship between the two. And when we did this, if you actually extended your epochs to be large, what we got is something that looks surprisingly like a sinc function. If we zoom in, we see that they actually do have these oscillations on both sides, right? It looks non-causal. So the interesting thing here was, well, this was just us comparing our spikes to the actual oscillations we already knew, the same recordings from the brain. And if we were to zoom in on those spikes specifically, we see that for the most part, they were made up of two things: spikes, just individual small spikes, and bursts. And so this is an important point, because those bursts, we hypothesized, would have a lot to do with why we were seeing this effect, right? If you have these very, very strong bursts that are related specifically to your oscillation, maybe that could explain some of this. So as you guys are aware, you can use your unit impulse, and if you put it through an ideal filter, you get a sinc function. Well, if you identify your spikes, you can consider those like unit impulses, right? You could model each spike as a unit. And if you put that through an ideal filter or a rectangle, right? So an ideal filter means that here's your brick wall. You want to make sure that these sides are straight, rectangular functions, right? So if you actually put this spike train through your ideal filter, what we got back was this plot. And what we see is that that looks pretty similar to the plot in point A, or in part A, right? It looks very similar to what your brain what was actually recorded from your brain. And if we made a power spectrum of this as well, we see that we're really recovering that dominant frequency that we expected. And we also, just as a double check, we rearranged our spikes. We said, okay, keep the same number of spikes that we've identified, but now put them in whatever randomized series you want. And we put it through our filter. And what was really nice as a check was that the structure was broken. There was no seizure to be seen anymore. And so what this really came down to was suggesting that, hey, not only can we see this effect and it's representative of what we're seeing in our ECoG, but there's something really special about the structure of how the neurons were firing, how we were identifying these spikes. And our hypothesis was that it had something to do with these bursts, right? These highly stereotyped bursts. So to explore that, specifically, we went through and we said, all right, we have our seizure and we know where all of our identified spikes are. What happens if I take away a certain percentage of those randomly? How many spikes do I need to remove before I ruin the structure? So this is another form of checking it. The same way that we did the randomized bootstrapping, you can remove a certain number of your units here. And so what we did was to remove them in 5% increments. And what we saw is that it took us all the way to 85% of these before you actually lost that relationship. And so not only were these bursts really driving it, but it was really, really stereotyped bursting, right? That was causing this effect. And so how do you check this? Well, the easiest way is to say, okay, if I do think that it has something to do with the bursting and it has something to do with how structured things are, I can build myself a simulation. I can make myself a different spike train, just using those parameters that I think are important. And so that's exactly what we did. We built our own little pretend seizures, looking at the multi-unit activity. And what we got back was pretty neat. So remember, this is what the clinicians saw. This is what happened when we put the identified spike train through our BrickWall filter. And this is what we got when we just used those parameters and built our own spike train. And so using the key clues that we identified. And so we thought that was pretty neat, that you could actually describe this relationship just based on a few key features of your spike train. But this is all well and good. At the end of the day, we wanted to take this a step further and say, okay, well, how physiologically could we possibly represent this? Can we come up with any models that sort of hypothesized on where all of this is coming from? And so my work before this, where we had really focused on those spike triggered averages and trying to understand the relationship between the spikes and the ECoG, had led us to this idea of a forced oscillator. So in this case, we have this forcing term, W. And what this forcing term is really representing is that wavefront, right? That big high activity that was right at the edge of the core recruiting. We're going to say that that had a lot of influence on your neuronal networks. So we're just going to say our basic brain network has an excitatory component and an inhibitory component, and they're connected, right? That makes sense. You have seen that possibly. I don't know which classes you guys have had at this point, but the brain has excitation and inhibition. And if we have this activity, we could activate that EI network and probably get these oscillations. So the question was, could having this intact inhibition here, right, explain our oscillations when this is that core area, really forcing everything, right? Really activating everything. And so we built this form of a model where we actually had a more physiological basis. And again, we fed in that spike train, the spike train from our seizure as our forcing term. And so we said, okay, I want to see what happens in my network if I give it a whole bunch of really structured spikes. And what we got out was, again, something that looked very, very similar to our seizure. And not only that, but if you actually looked at what the relationship was, again, we got something that looked like this sync function. And so what this really reiterated in comparison to our previous work was that, yeah, you know what? This could be described. There are these really stereotyped spiking, and that can really describe a lot of what we're seeing. But, and this was the really cool but, if we go back for a second, I told you guys that in this little yellow portion, you don't really have inhibition, right? It's pretty structured. That's where you're getting that stereotyping. In order to get those oscillations, though, we had to keep our inhibition out here. And so this really suggested that there has to be a dual role for inhibition. In your forcing term, you have to get rid of it so that you can make that spike train exactly what it needs to be. But on the outside, in that bigger, broader penumbra, right, where we're recording, in order to get those oscillations, you had to keep your inhibition intact. And so that's kind of a crazy juxtaposition, that in one case you have to have it, and in one case you have to have it be canceled out. So that was how we brought everything together. That is the very short summary of that paper. So can I take any questions? Or comments? Or you can just admire the cartoon. I thought he was funny.
I can see that Derek is trying to ask something, but we don't hear you, Derek.
Oh, it looks like there's also a chat box. Hang on. Yes, Derek, if you were speaking, no one could hear you.
Yeah, no. Otherwise, Derek, why don't you type in your question in chat? We still don't hear you, so why don't you type in your question in chat?
Sorry. Oh, great question. So the question says, for part C of the figure, what does pseudo ECoG mean? Let me just go back to, I think, the slide that you're talking about. And of course, now my screen is frozen and doesn't want to go to that screen. Cool. But I can at least explain. So at one point, we were interested in, we had our ECoG recordings, but we also wanted to know what exactly would be the representation at the same point as our microelectrode array. And so we just averaged the activity across that array to get something that was equivalent to our ECoG, because the ECoG was a representation of a population of neurons. So we just did the same thing for our microelectrode array so we could get something comparable. Also, I will stop sharing and reshare so I can actually show you guys what I'm talking about. Okay, so hopefully this will make more sense. The microelectrode array here obviously didn't have an equivalent. So if you average your activity across the whole array, you could get an approximate pseudo ECoG. Any other questions I can answer?
I have a question.
Yeah.
So I know you're looking, like you did, you said something about having like, for the simulation, those really strong, like potentials or signals that would end up creating this sort of seizure. Is there like a, what about for non-seizures, as in, like, is there any time where the simulation wouldn't like result in a seizure? Do you kind of get my question? I don't really.
Yeah, so it's only under certain parameters that you get the seizure. So we actually, I didn't put it in here, but we went through a whole bunch of iterations looking at whether or not it was specifically bursting, and if you had bursting, how much noise or jitter could you have between the spikes, and what sets of those parameters actually describe your seizure best. So there were plenty of times where you don't get something that's exactly like the seizure, but we were specifically interested in figuring out when it would. Does that answer your question?
Yeah, I think so.
Okay. Any others or not?
I actually have a question on that same figure as well.
Sure.
I think specifically panel B. So
Yes.
you guys removed all, so you removed each of the spikes until the correlation like drops significantly. Is that the right way to interpret it?
Correct. So what we were really doing is saying, okay, of my total spikes, let me take away a certain percentage of those. Randomly pick whichever ones you want, but remove a certain percentage. And what we did then was to compare the relationship. So we took the cross correlation between that and the original recording, right, with all of the spikes intact, and said, how similar are these two? And it took until we hit 85% of them removed for you to get a significant decrease in correlation. So that's what's shown here. And if you, this should be 85-ish. I think it is 85%. We just chose whenever it drops below 0.9, so below 90% relationship there.
Thank you.
So I think that Tara deserved a virtual applause. And thank you very much for giving this talk. Just for your information, Tara won a prize with this paper as most innovative paper of, what was it, 2018 or 19? I'm kind of starting to lose track of it. But
the paper was from 2018, but the award is for 2019. So I don't know if no one published the next year or what.